Hi, my name is David Collins, and I'm a PhD candidate in the lab of Dr. Marina Kalyuzhnaya in the Cell Molecular Biology program here at San Diego State University. And I'm going to talk to you about my research on how metals impact bacterial structure, cellular performance, and metabolism. These microbial outputs have many impacts on the environment, as well as the contemporary struggle of global climate change. Now, across many domains of life, it was noticed a common feature of an inefficient form of metabolism called overflow metabolism. And this was seen in things such as cancer cells as well as E. coli. This form of metabolism is unique because it's an inefficient breakdown of substrate in which 6 carbon sugars are broken down into pyruvate, which then normally, under respiration conditions, will be broken down into carbon dioxide, as well as lots of energy. But in an overflow metabolism, even under aerobic conditions, this breakdown is not complete. Oops, I've already messed up. Now, the key difference between these two outcomes is their locations. Cancer cells stop depending on their mitochondrion, and E. coli's lack of mitochondrion entirely. And as a result, carbon cannot be broken down because the electron transport chain is not being used as much. Fermentation occurs in the cytosol and relies on enzymes which are less energetically costly for the cell. And what are the consequences of these outcomes, and how could we test it in the laboratory? And this is through another model organism I'd like to introduce, the aerobic gamma-proteobacterial methanotroph methylo2V microbium alkylophyllum. Now, this microbe is able to take the greenhouse gas methane as its sole carbon and energy source, from which it builds all its biomass. Conveniently, we can grow these microbes in different conditions in which cellular structures can be altered. Now, this is through trace metals such as copper. When these cells are grown without copper, they very much resemble E. coli. However, in the presence of copper, they develop an extensive intracytoplasmic membrane network, and in this way they resemble the eukaryotic mitochondrion. Now, in these two different states, we can grow them and measure cellular performance to determine how their metabolism changes. So we feed these microbes methanol, and then we measure different cellular outputs. One, membrane area. The intracytoplasmic membrane network causes much more membranes to be developed in the presence of copper compared to without. Additionally, growth rate, which we call mu, is higher in the presence of copper. Under the presence of copper, these cells also consume more methanol and oxygen. Additionally, cells in the presence of copper produce much more carbon dioxide. Now, the enhanced amount of real estate of membranes in these cells under this condition allows them to maintain their high growth rate without switching to less energy efficient metabolism. Additionally, these microbes have less efficiency compared to without copper in converting carbon into biomass. So in a way, these microbes are burning through substrate using the electron transport chain and respiratory machinery compared to growth conditions without copper. Now, the way microbes and complex systems such as cancer function is understood by their cellular outputs and their metabolism. And how microbes use their substrate is important as we develop these organisms into biocatalytic platforms. So as Earth begins to have increasing amounts of carbon dioxide and other greenhouse gases in the atmosphere, understanding the metabolism of these organisms allows us to use greenhouse gas capture, which can then be fed to these microbes and use them as platforms for next-gen materials, as well as value-added products. And with that, I thank you for your attention, and that concludes my presentation.